Okay, we are picking back up where we left off, talking about the two different types of cells there are in life. So we can divide all of life, all of cellular life, into two different basic types: the eukaryote and the prokaryote. Eukaryotic cells versus prokaryotic cells. The prokaryotes consist of organisms called bacteria and archaea. These are single-celled, teeny tiny organisms. Bacteria you're aware of. They're the things that make you sick sometimes, even though the vast majority of bacteria are not pathogens. They do not make you ill. There are some pretty famous bacteria that make you ill, like, Streptococcus pyogenes, which causes strep throat. Anyway, there's also the archaeans, which are prokaryotes. So archaea. Archaea consist of teeny tiny single-celled organisms, but these are hardy little guys. When you look at a hot springs over at Yellowstone, and it's bubbling, boiling hot, full of sulfur and other poisons, and you see these gorgeous colors in there like bright blues, those are caused by archaeans living in there, living in those harsh conditions. There are archaeans that live in Antarctica. There are archaeans that live in the volcanic soils or around volcanoes, near the really, really hot parts. These are organisms that thrive in horrifying environments that kill other life. Archaeans are called extremophiles. They're the 1990s Mountain Dew commercial organisms of the world. The extreme organisms. Leave me alone. Anyway, then there are the eukaryotes. Eukaryotes are everything else. They're the single-celled protistans. Then there's the animals, plants, and fungus among us, the fungi. All right, so these are two basic divisions of all life, but they're not super different because they actually have several shared features. So let's take a moment and talk about the features common to all cells, no matter whether it's a prokaryote or a eukaryote. One of the most important features is the cell membrane, sometimes termed the plasma membrane in your book. The cell membrane surrounds the cell, right? Without it, there is no cell. It is semi-permeable, which means it allows some things to enter and prevents other things from passing. So, whereas oxygen might pass right through the cell membrane because cells need oxygen, damaging ions might bounce off. It might find it's impermeable to damaging ions. And at the same time, if this is the inside of the cell, there's some vital nutrients in here we don't want to just leak out to the external environment, you'll find that the plasma membrane, the cell membrane, is impermeable to those. So it is semi-permeable. Lets some things through, blocks others. So the cell membrane itself is pretty much, you gotta have it to have life. Without a cell membrane, there is no life. So that is a sine qua non thing, without which nothing. Sine qua non. I think that's it's Latin right there. I learned it from Battlestar Galactica because I get all the important life lessons from pop culture. The cytoplasm is the next shared feature, also called the cytosol in your book. Cytoplasm is important. This is what fills in the cell, right? That cell membrane surrounds the cell. Well, it's not hollow inside the cell. In fact, it is filled with a particular jelly-like fluid called the cytoplasm. Next up, we have the organism's genome. So we have, I edited that. Anyway, let me edit that again because I liked my edit that when the computer crashed and lost other things, it must have lost that, which is just awesome. Yes, you're going to see me type this, and then I'm actually going to say it out loud for the sake of redundancy and repeating myself. So, the organism's genome is made of DNA. The genome is its genetic, the total of all its genetic information. That is the genome. All of the organism's DNA in a cell makes up its genome. DNA is organized into genes, and one gene equals one protein. So that is the basic organization scheme of the genome, of the genetic material of an organism. And whether it's prokaryotic or eukaryotic, if it's cellular life, it has a genome made of DNA that is organized into genes. And that's important because each gene has the instructions for a protein. And since your DNA is just a giant instruction manual for making proteins, we got to have some place to build those proteins. And that happens to be at the ribosomes. So if you remember from chapter 3, ribosomes are actually constructed of RNA. So, and these are the structures where proteins are made. They're the site of protein synthesis. So they use the instructions from the genes and make proteins. All right, so those are the commonalities to prokaryotes and eukaryotes. Let's talk about the differences between prokaryotes and eukaryotes. Well, for one, in a eukaryotic cell, DNA is found in what we call a nucleus. A nucleus is sort of a membrane surrounding, a chunk of cytoplasm, so sort of like a little membranous sac. And inside that sac is the DNA. So the nucleus is a structure containing all of the DNA. That structure happens to be membrane bound. Eukaryotes have their DNA in the nucleus. Prokaryotes do not have a nucleus. Instead, prokaryotic DNA is sort of loose in the cytoplasm. In fact, another important difference while we're talking about DNA, eukaryotes have linear DNA. That means that, in effect, linear means a line. You can think of eukaryotic DNA as sort of like a giant ball of string, except instead of one continuous piece of string, you've cut it into multiple different links that are still wound up together into a ball. And if you pull one of those individual lengths of string, you have a beginning and an end. That is what it means to be linear. There's a beginning and an end. Eukaryotic DNA is linear strands. Prokaryotic DNA is not linear. In fact, it is circular. And we see that the genetic information of a prokaryote is circular. In fact, you can even have two sets of DNA, and one is the sort of the main genome that's circular, and then you can also have an additional circular plasmid that carries sort of spare genes that help it do other things. For instance, the superbugs like MRSA that you hear on that House MD show. MRSA stands for methicillin-resistant Staphylococcus aureus. Staphylococcus aureus is a type of bacteria. Methicillin is an antibiotic, so it is resistant to an antibiotic. That gene happens to be carried on a plasmid, so spare DNA that gives it new properties. Eukaryotes have what we call membrane-bound organelles. These are little structures or sacs that are, well, surrounded by membrane. In fact, the same stuff that the cell membrane or the plasma membrane is made out of is the same thing that surrounds the organelles. So an organelle is sort of like a little specialized structure. And each organelle performs its own specific job in the cell. You can kind of think of it like an office building, right? And each of the different workers who has his own specific job is a different organelle. There's an organelle for the factory floor. There's an organelle for the warehouse workers. There's the organelles for, the people who make things. So there's organelles for everything. In fact, remember how I said the nucleus is made of membranes? It's sort of a membrane sac, membrane bound. Well, the nucleus itself is an organelle. Prokaryotes don't have a nucleus because, well, prokaryotes don't have membrane-bound organelles. So prokaryotes have no membrane-bound organelles. Eukaryotes are much, 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 much bigger than prokaryotes. So on the other side, I have very, very, very small prokaryotes. And then this is just reiterating what I said before, except I accidentally forgot to write archaea there. So eukaryotes are the protists, animals, plants, and fungus, and bacteria and archaeans are the prokaryotes. Here's a great example of a difference in size between them. So this is an average eukaryote, and here's an average prokaryote. You can see that prokaryote is vastly smaller than the eukaryote. And if we look at that eukaryote, you can see all these little specialized structures in here. Here's an organelle. This is an organelle. This is one. Here's one. Here's an organelle. So all these specialized organelles, including the nucleus that contains all the DNA. And then over here we have our prokaryote. 0 organelles, just, our circular genome here. Not a perfect circle because, well, it's life. Life is messy. But it is circular, as in non-linear. There's no beginning and end. Then we have the cytoplasm filling it in. Then we have all these little dots, which are individual ribosomes, and then the black cell membrane. So prokaryotes are, in general, much simpler than a eukaryotic cell. So pretty cool. All right, let's go into some details about that plasma membrane. Plasma membrane is made up of what we call a phospholipid bilayer. And as we said earlier, it is semi-permeable. Semi-permeable means it allows the passage of some things, like oxygen, water, or sodium, but it blocks others. For instance, damaging ions can't enter the cell, and necessary things like important nutrients can't leave the cell. So here we go. This right here is a phospholipid. So a phospholipid is a type of fatty molecule in which we have 2 fatty acid chains attached to a phosphate head. Now what's cool about phospholipids is whenever you get a bunch of them together and sort of shake them up and throw them in fluid, they organize themselves into this funky cool structure where you have all of the phosphate heads hanging out together facing outward, whereas and then you have this interior here where you have the 2 fatty acid chains or tails. So this is a phospholipid bilayer. Bilayer because, well, here's one layer of phospholipids and here's a second layer of phospholipids. Bilayer. So this is a phosphate head. These are the 2 fatty acid tails. And the fatty acid tails are hydrophobic, whereas the phosphate heads are hydrophilic. This is one of the reasons why they naturally orient themselves without any help or instructions. When you throw phospholipids together in fluid, they will naturally line up and orient themselves so that the fatty acid tails, the hydrophobic tails, face the interior of the bilayer, and the phosphate heads face the exterior of the bilayer. And this is simply because the heads come in contact with your fluid medium, your water, and those heads are hydrophilic, whereas the hydrophobic tails are now protected in the interior, the hydrophobic interior. So, pretty funky. What's neat about this plasma membrane is that you have this giant nonpolar hydrophobic interior, which means things that have sort of a charge to them generally don't pass through the cell membrane. They'd have to be able to get through the nonpolar region, and well, charged things and nonpolar things, they don't mix. So instead you have transport proteins, and they allow larger molecules that are too big to just diffuse between the phospholipids and charged molecules to pass through the plasma membrane. So a functional cell membrane is not only a phospholipid bilayer, but a whole bunch of embedded compounds like transport proteins, cholesterols, carbohydrates attached to proteins, making these glycoproteins. So receptor proteins. So some pretty fancy stuff. Like I said, oxygen, O2, can just go straight across because it's small enough. Okay, so while we're talking about the plasma membrane, let's mention the fluid mosaic. This is sort of a term for a model of the plasma membrane. What is a plasma membrane? Well, it's a phospholipid bilayer. But how could we describe its structure, its features? We use the term fluid mosaic. Membranes are not, in fact, rigid. They are very fluid. They're sort of in constant flowing motion. And that gives them sort of a fluid behavior. Membranes are fluid. And then embedded in them is a mosaic of proteins, carbohydrates, and cholesterols. So that's why we have mosaic. A mosaic, if you take a look, if you want to Google the word mosaic, you'll see a bunch of probably Roman pictures made up of colorful tiles all glued together to make a larger picture. So a whole bunch of small things put together make a larger structure. And so the mosaic of proteins lends a larger certain functionality to your cell. So that's why we call it a fluid mosaic. So membranes are held together by the attraction between the hydrophobic phospholipid tails. It's not a particularly strong interaction, by the way. So lipids and proteins are able to shift about laterally in the membrane. So if this is, you looking head on at my membrane, and here's my protein, it's going to be sort of moving about laterally. It can move all throughout my membrane, all laterally around, kind of like people sort of pushing each other and elbowing each other around as they move through a crowded concert. So let's take a nice little video.
Between the lip.
So we're zooming in on a cell. This is the membrane. We're going to get nice and close in our 90s-tastic video. These hexagons here, those are the phospholipid heads, and then here's the tails. We're showing how it's fluid, right? And we're seeing how if you bonk it hard enough, you'll knock them out of place, but because of the attractions between the tails, they come back into place. So phospholipid bilayers self-arrange. Now we're embedding some important stuff in there, some carbohydrates, some cholesterols, and now we need to add some proteins to give it certain functions. So there we go. There's some proteins. And now we're going to start transporting molecules around through these proteins. That's going to give us some nice little features to our membrane. Make it nice and functional, enhance the permeable nature to it. And now let's take a look at how proteins can shift about laterally in my fluid mosaic. So now you can see these proteins sort of rotating around and moving laterally. So pretty fancy. So while we're talking about things, let's mention the cell walls. Cell walls are found in plant cells. You also see a certain type of cell wall in fungus and in bacteria and archaeans, but this is 105. We'll talk about fungus later if we have time, and we're not really going to touch too much on bacteria and archaeans. So when I say cell walls, for this lecture at least, I'm referring to plant cell walls. Plant cell walls surround the outside of the plasma membrane, and they lend structure to it. They provide strength and shape. And they are made from cellulose, which is a carbohydrate. And it, in addition to providing, strength and shape, it also armors it against the outside world. So very, very important, especially since plants have no skeleton, so to speak. So it's the cell walls that help them resist gravity. So there you go. This is a cellulose cell wall. The pink is the plasma membrane of the cell, and the green is the cellulose cell wall. Again, falsely colored to enhance contrast. But still, you can see these arch structures here that have a sort of an inherent resistance to pressure on them. So that's a very strong structure. Provides armor, strength, support. Very cool. So what you find outside of the membrane on a plant cell is pretty neat. Armor, like you'd find on the weirdos at the Renfest. If only I could be one of those weirdos. But armor's too expensive. Anyway, what's neat about plant cell walls when we look at it under microscope like this is that they do such a great job of providing armor, of isolating the cells, that you actually have to have canals between plant cells in order for them to be able to transfer nutrients and chemicals between one another. Because there's no blood system to supply, local cells with blood supply. This is a plant, man. So instead we have these little canals in the cell walls. And that basically means that this cell, and this cell share one continuous cytoplasm because of those little canals called plasmodesmata. That's not the important part of this picture. What's important is the actual image of cells surrounded, armored by their cell walls. Now, plant cells aren't the only things that have something really cool going on outside of the plasma membrane. In fact, multicellular animals with tissues have the extracellular matrix. Extracellular matrix is pretty sweet. That's where you go outside the cell in order to learn kung fu, right? Not delivered well enough? Perhaps I should say it in in a Keanu Reeves voice. I know kung fu. Meh. Moving on. The extracellular matrix is actually pretty cool because it's this giant sticky mess of like sugars and fats and collagen and other stuff that helps glue cells together. So it's between animal cells, and it's this sticky stuff that's secreted by the cells and keeps them anchored together. And because they're sort of anchored together, they're near each other, so it helps in communication between cells. It helps to glue the cells together so that they, stay in a particular shape or area. And of course, it helps to hold their cell shape. So it's pretty neat. Here is green stuff, and this green stuff is extracellular matrix. And the orange stuff is individual cells. And so each of these cells has been secreting the green stuff to hold them in place and help them communicate, right? Because they're stuck in all this green stuff. These two cells here are right next to each other for the duration. And so when this cell releases a chemical that this cell can respond to, it's very easy for them to communicate, for them to share chemical messages. And then it helps them support their shape, helps keep the cells all together in a layer. So extracellular matrix, pretty cool. plants don't get to have all the fun. Alright, so we'll pick up with the next video and we'll finally go into the anatomy of what is inside the cell. And we'll be focusing on eukaryotes. So, see you in the next video.